immature? 92. How many immature, oh my God, I'm so sorry. How many DNA molecules are present at the end of G2 phase in an immature sperm and an immature oocyte? 92 also. Nothing happened between here and here in terms of the DNA molecules. So make sure you know these numbers, right? Like that's just something I could ask, like comparing mitosis, meiosis, and the number of chromosomes at different stages. Okay? All right. So we are now going to go into prophase 1. Prophase 1. What am I, whenever you see P1, right? You know what, I'll just do regular instead of like Roman numeral, I'll just do P1 like that. So whenever you hear P1, you prophase 1, telophase 1, metaphase 2, you know we're talking about meiosis. Yeah? Okay. So what happens? Well, some of the things that happen in regular prophase, you know what I mean when I say regular, mitosis, right? And prophase with mitosis are also the things that happen in prophase 1, right? And so, you know, the DNA molecules, what format are they in here, you guys? What type of format? Chromatid. So the chromatin will do what? Condense into chromosomes. So chromatin does condense into chromosomes. And, right, so the sister chromatids become attached at their centromeres, right? So let me draw that, right? So chromatin condenses into chromosome. This is an immature sperm or oocyte, and the sister chromatids become attached at their centromeres. The sister chromatids become attached at their centromeres. Okay. Now, I'm going to draw this here. Another thing that happens, notice how I drew this here versus here. See the sister chromatids, these sister chromatids, they're not close to these sister chromatids. These sister chromatids, they're not close to these sister chromatids. Notice what happens to these. The sister chromatids, right, of these homologous, the red and the blue here, you guys, the red here versus the blue here, what are these to each other? They're not identical. I mean, the red is identical, the blue is identical, but the red here and the blue here, they're not identical, but what are they?
Homologous.
They're homologous to each other. They're very similar. So basically, the homologous pairs, each made up of two sister chromatids each, right, come together in this process. I'm not writing this down, but this is like actually unique for prophase 1, right, compared to like prophase, right, and it's in my PowerPoint. So the homologous pairs, and I could keep drawing more of them, I'm just not going to. Each made up of as two sister chromatids each, right, come together in a process known as synapsis, not synapse, synapsis, right, forming these 1, 2, 3, 4 like structures called tetrads. Right, so these are some key terms, right? So homologous pairs of chromosomes, each made up of two sister chromatids each, right? So 1, 2, 1, 2, right? Come together in a process. This process is known as, this coming together is known as synapsis, right? And they form this structure known as tetrad, because tetrad means 4. Is this clear so far? Yes? Okay, so that's unique. That's a unique event that happens. The chromatin condensing into chromosome and the sister chromatids being attached to one another first, right? That happens in prophase as well, right? So that's not unique, but I do want you to know that, right, for prophase 1. I mean, some other things, what else happens? I mean, nucleolus disappears. I don't even have them listed for prophase 1. Right, so if I asked you about prophase 1 to describe it like on an exam, like if you added nucleolus disappears, great, but if you didn't add it, I wouldn't take points off for that. Right, because that's, I mean, yes, it happens, but that's not the like main thing I want you to remember for prophase 1. Yeah. Does meiotic spindle, what's it called, appear? Yes, so we have these centrioles, these two centriole pairs, and we have meiotic spindle, right, growing out of them, these meiotic. Why am I calling it meiotic all of a sudden instead of mitotic? Because it's meiosis, right? But these are, you know, these guys are those polar microtubules, the, what's it called, the kinetochore microtubules, they start growing out, right? And eventually what's going to happen is they move to opposite poles of the cell, right? And eventually, they're going to be attaching also to our chromosomes, but differently this time. So they're going to be unique. Okay. So I think in my description, I do say the centrosomes, centrioles move to opposite poles of the cell and produce meiotic spindle fibers, right? So that's something I do want you to know that detail, right? We already talked about the chromatin condensing into chromosomes, the sister chromatins being attached to one another. Eventually what's gonna happen is this, and that's important to know, this is gonna start disappearing. You know what, I'm gonna also draw another prophase one, I guess, and then anaphase. We'll do it like this. God, we'll see. Okay, so this is still prophase one. This is still prophase one. But I'll do the centrioles here at the opposite poles. The nuclear envelope has now disappeared. The meiotic spindle fibers here. Now, one thing that is unique, another thing, not one thing, because I already talked about a couple of different things, but another unique thing that happens in prophase one, due to these tetrads coming together via synapses, is this thing called homologous recombination. Okay, so let me draw what I mean. I guess I'll put this like this. I'm gonna just do it already as if it has happened. Okay, see how I like drew this weird? Okay, this is kind of weird to draw this. But what happens is when these homologous pairs of chromosomes come together, the sister chromatids that are like closer to one another, right? This sister chromatid of this homologous, of these two homologous pairs, these two are not identical, but they actually exchange, they end up exchanging genetic information. Some of the genes that are on this sister chromatid actually moves all, like flips, for lack of a better word, and gets onto this sister chromatid, and some of this flips and gets onto that sister chromatid. So you have basically exchange of genetic information between the homologous pairs. Between the homologous pairs, you have an exchange of genetic information. Okay, that's known as homologous recombination, because you're like recombining genes, right? And you're doing it between the two homologous pairs. Something, so basically the genetic information crosses over. It's known as, the scientific term, the non-scientific term is called crossing over, but please use the term homologous recombination. And basically, and be able to describe it, right? That your homologous pairs of chromosomes exchange genetic information. Okay, any questions? Okay, so let me then do this guy, maybe I'll do this guy on that side. Just a little bit. Okay. What else happens? Meiotic spindle fibers attach. These purple ones are those kinetochore microtubule meiotic spindle fibers, or you can just simply, for prophase one, you can just call them meiotic spindle fibers. They attach to your tetrads, to your homologous pairs. From both poles of the cell, but would you say they attach to sister chromatids from each pole of the cell? You wouldn't, right? If you look over here, like, does the purple go and attach here? It can't. It doesn't. It cannot. Right. So it's attaching the meiotic spindle fibers, more specifically these kinetochore microtubules, are attaching to these tetrads from both poles of the cell, to these homologous pairs from both poles of the cell. So you wouldn't say they attach to the sister chromatids. Like notice this image versus this image from prophase of mitosis. Here, the kinetochore microtubules and mitotic spindle fibers are attaching to the centromeres of each of the sister chromatids of each sister chromatid. Are they doing it here also? The centromeres of each sister chromatid. No, this is a sister chromatid, another sister chromatid of that sister chromatid pair. This is here, right? So what we're attaching to, what these meiotic spindle fibers are attaching to, are the tetrads, right, from each opposite pole of the cell. Or another way to say it is to the homologous pairs here and here. Is that clear? And then what do the meiotic spindle fibers do? They do that whole tug of war thingy, right? Just like they did here. And so what will happen next in metaphase 1? What will be brought to the center of the cell equator? The tetrads will be brought to the cell equator. I kind of already, you know what, I'm actually gonna go ahead and draw, well, because it's kind of already in the center, so this image and metaphase 1 is gonna look exactly identical. I would have like drawn this a little bit here and this here, right? So make sure you maybe I'll do some like recombination already here. Let's do some recombination here. Homologous recombination is happening. I want to be consistent, so here. So so here's homologous recombination, the swapping of genetic information between these homologous pairs, swapping of genetic information. How do I do it here? I do it here. Swapping of genetic information. Swapping of genetic information. Okay, so then I'll change this to M1 because otherwise it's the same picture if I keep drawing it right M1. So this is metaphase 1. This is metaphase 1, where who is aligned at the cell equator? Our sister chromatids aligned at the cell equator. These are sister chromatids. These are sister chromatids. These are sister chromatids. These are sister chromatids. Are they in the center? No, right? Because this is sister chromatids in the center, right? This right here from metaphase of your of mitosis, this is sister chromatids aligned at the cell equator. Who is aligned here in metaphase 1 of meiosis? It's your tetrad. Tetrads, you could say tetrads are aligned at the cell equator, or the homologous pairs are aligned. Like these guys are homologous pairs to one another. The homologous pairs are aligned at the cell equator. How did they get there? Who made them go there? Those meiotic spindle fibers. Okay. Any questions so far? Okay, so let's review what happens in prophase 1. So I'm going to pull this down and talk about some of the unique things that happen in prophase 1. Because some of the things that are happening are not unique, like